Okay, this is a video to go over question 6 of the Foundation Chapter 4 Biology End of Chapter test. It's a very long question, it's 17 marks and it's got loads of stages, so I'll probably break it down into two parts. So, complete the equation for photosynthesis, draw a ring around each correct answer for 2 marks. So, always the same, the two reactants are carbon dioxide and water, and they react to form oxygen and glucose. So 1 mark from water, 1 mark for glucose. Keep reading. Some students investigate the effect of light intensity on the rate of photosynthesis in pondweed. The diagram shows how the apparatus the students used. So we've got some pondweed. Do bear in mind the scientific name for pondweed is Elodea. You've got a boiling tube and a beaker. You've got water held at 20 degrees in the beaker. You have got a lamp which you can change the distance it is away from the boiling tube. The closer the lamp is to the pondweed, the more light the pondweed receives. The students place a lamp at different distances. D, from the pondweed they counted the number of bubbles of gas released in a minute for each distance. A thermometer was placed in the glass beaker. Why was it important to use the thermometer in this investigation? So we have to say 3 things because it's 3 marks. So, I've said temperature is a limiting factor for photosynthesis at low temperatures. Molecular collisions are less, and this means this prevents photosynthesis happening quickly. At higher temperatures, enzymes are denatured, and this prevents photosynthesis from happening too quickly. There should be another O on that too. The students need to control the temperature to get valid results. So let's have a look at the mark scheme for that one. So to measure the temperature, to check that the temperature isn't changing, the rate of reaction changes with temperature. Temperature is a variable that needs to be controlled. So any 3 of those 4 points. So thermometer is there for the temperature. The thermometer shouldn't change because changing the rate of changing the temperature will change the rate of reaction, and just clarifying that temperature is a control variable. Good. Moving on. How much longer have we got? About halfway. So students counted the bubbles 4 times at each distance and calculated the correct mean value of their results. The table shows the students' results. So we've got a gap here for 40 centimeters. So I have realized I think I have made a mistake, but it's very interesting about this. So you can see the 4 repeats, 3 of them are around 10 and 1 of them is right up at 30. So I have just added them all together, so 30, 10, 9, 11, and divide them by 4, and this wouldn't get me 2 marks. It would only get me 1 mark because 30 is an anomaly. So you should have done 10, add 9, add 11, which itself makes 30, divided by the 3 good, valid repeats you've got, which would give you 10 bubbles per minute. So the 30 shouldn't be included. So this answer I've got here would only give me 1 mark. And I'm going to end this video here. There'll be a part 2 to follow. Okay, thank you very much.